Hey guys, in this video, I'm talking about the ABO blood group system as taken by Dr. Kingsley, hematology lecturer. Okay. For starters, let's discuss what blood group is. Blood group or blood type is defined as a classification of human blood based on the presence or absence of antigen on the surface of red blood cell. Now, I'm sure you might be wondering what the meaning of antigen is. Let's go further. Antigen is a substance that induces an immune response. Now, in this case of blood grouping, which is not just talking about just anything, it refers to specific protein present on the surface of red blood cell membrane that cause blood reaction, which lead to the destruction of red blood cell when it come in contact with blood that is incompatible with it. Now, having understood the basics, let's discuss ABO blood group system. The ABO blood group system is a blood group system in which classification is based on the presence or absence of the A or B antigen on the surface of red blood cell membrane. It was discovered by the Austrian scientist Karl Landsteiner in the year 1901.
ABO antigens and antibodies are produced at different times in life. Now, the antigens are produced from birth or even before birth, but the antibodies start being produced at about 3 months in life. Sometimes it can be later, so that's it. This is the reason why blood grouping is only done on babies as from 6 months of age, and reverse blood grouping is not done for them. Now, ABO antibodies are transferred passively from mother to child within these 3 months through breast milk.
Now, let's discuss the blood groups under this ABO blood group system. We have four. They are the A, B, AB, and O blood groups. Now, please note, the blood groups are represented with positive or negative is when you combine the versus with the blood ABO blood group system. Now you get that. ABO incompatibility. Now, ABO incompatibility refers to a phenomenon or occurrence in which blood of different and incompatible blood, that is blood that do not match, mixed together, whether it be in vivo or in vitro. In vivo, inside the person's system; in vitro, in the test tube or on the slide. This mixing causes blood reactions, which lead to the destruction of RBCs within the circulatory system of the patient or the blood itself, whether you put it in vivo or in vitro, which can have adverse effects on the well-being of a patient. It can even lead to death. Now, the reaction may be seen as clotting of blood or hemolysis. Now, ABO incompatibility can be seen in two particular cases. One is blood transfusions, where you mismatch blood transfusion, and in rare cases you can see it in case of newborns and fetuses when the blood group of the mother is incompatible with that of the child, especially in cases where the mother's blood group is O and the child is A or B. Now, this is a chart of the various blood groups on the ABO system. You can pause the video here to observe and study them. So let's continue. ABO genetics. This basically talk about the genetic control to determine the blood group of a person. ABO blood type is determined by a single gene, the ABO blood gene, and the gene has three different alleles or forms, which are the IA, which determines the blood group A, IB, which determines the blood group B, and small letter I for blood group O.
Alleles of the ABO blood group system are expressed in pairs. Thus, two alleles are required to make a person's blood group. Now, knowing this, we have the following genotypic expression. That's how the person's gene can look if you are looking at the genetic disposition of blood group. Now, we have AA and AO, which can be disposition for A, BB and BO, which can be disposition for B, AB for AB blood group, and OO for O blood group. Now, talking about the biochemical nature of ABO blood group, it involves a complex interaction with carbohydrates, proteins, and lipids.
The gene encodes a glycosyl transferase, which is an enzyme that transfers N-acetyl D-galactosamine for antigen A and D-galactose for antigen B to the non-reducing end of glycans on glycoproteins and glycolipids.
The primary structure of this antigen sequence is either a glycolipid or glycoprotein with an oligosaccharide precursor sequence, which is a carbohydrate sequence, and one or more specific sugar molecules attached in specific location. Now, if you look at the picture that was given, it basically talks about a situation where neither of both parents are blood group O, but they ended up giving birth to a child of blood group O. Now, the reason for this is, remember when we talked about alleles that they come in pairs. Now, if you observe both parents have um, a recessive O gene because they have AO and BO respectively. Now, looking at this, we can see that A and B both have a codominance relationship. That's in a situation where both are present, they will both be expressed in the individual. And O blood group can be expressed in this case when the O allele of both parents come together to form the individual. I hope that you guys are able to get the explanation of the video so far. Look forward to future videos. Thank you.